Greetings, it's Mr. Picard here. And what I'd like to do today is do a science demonstration. And when I was thinking about it, I was trying to think, is this an experiment? Is it an investigation? What exactly, how do I begin? Because I'm hoping to show you something that gets you started on a scientific endeavor. And so how does science begin? And I was sort of thinking about that. What is the actual kind of impetus to get science started? And I'll tell you what, let's think about that just for a moment. Would you think about, how does science begin? I'm thinking, okay, so what came to my mind were some terms I think I learned in school, things like scientific method, hypothesis, correlation, causation. Maybe science begins with testing a hypothesis, or maybe observation. And I think probably some of those terms came into your mind as well. But I don't really think we have it quite right yet. And I looked up a quote that I'd heard from a science fiction and science book writer named Isaac Asimov. And I think I can remember the quote word for word correctly. And what he said was that the most exciting phrase to hear in science is not, "Eureka! I found it!" but rather, "Huh, that's funny." And I think what he meant by that, and the reason I think that applies here, is I think that is where science begins. When somebody notices something and says, "Huh, that's funny," meaning that's interesting, I wonder why that happened. And everything else sort of comes from there. So what I'm going to do is do a demonstration that I hope does that to some degree for you. So I invite you to watch carefully what I'm going to do. And then we'll think about where the science might be or how it starts. So I have a pitcher. And in this pitcher is a solution. And what I'm going to do is pour 7 different colors of liquid from this pitcher. But I'm not going to change the pitcher in any way. And so I'll show you what I mean. You know what? I'm going to predict which colors I'm going to pour. I think in this middle cup right here, I'm just going to pour purple. Look at that. Lovely, lovely purple color. I think you can see that. And let's pour a different color now. How about some red? Let's pour a red. Yeah, that looks nice. Same picture, purple, red. Well, let's try a different color. How about blue? How about some blue? That's a nice color, blue. And, Mason blue. I went to University of Michigan. How about some yellow? No, there you go. A nice yellow color. And what color could we do next? How about kind of violet? Would be nice. Not really purple, not really red. Let's take a look at that. Boy, that's a nice color. Violet. And how about some green? There's a nice green. And finally, I think we'll make kind of a pink, a little bit lighter red. Oh, well, that one didn't cooperate. We're going to let that one be. So I got one surprise, but everything else is pretty amazing. I've got 6 colors, different colors, out of one picture. And in some ways, I think this might be a kind of kiddies magic trick, but you know, I'm not a magician, I'm a science guy. And I do think this does qualify, however, for what Isaac Asimov would say, huh, that's funny. You know, they poured yellow from one picture and they poured violet over here. That's interesting. And I think this is a good place to start with some science. And I'm going to tell you how I did this, or what I did, I should say. But I'm not going to tell you why. I think that, you know, we're going to look into that later. And some of you may have seen this before. But inside each of these jars, I had a little bit of a chemical, of a compound, that when I mixed it with what I had in the picture, which was an acid-base indicator, it changes color based on how acidic on this end, or basic, also known as alkaline, on this end. And this indicator changes color based on acidity, or how basic something is. And in fact, the color of the indicator in water, and when it's neutral, is the purple, just like this one. And this one's also purple because I forgot to put the vinegar in it. But this one, I put lemon juice in, which is acidic, and it turned quite red. This one would have been more of a pink. This is a beautiful color of ascorbic acid, vitamin C. This again is just the water. The green is, the blue is a kind of a household cleaner, like a spray cleaner. This is ammonia, this beautiful green, and finally bleach on the end. These are all household things, you know, relatively safe. Please remember, if you do anything like this experiment, to be careful mixing or using household chemicals, because they, if not handled properly, can not be healthy. Especially, you never want to mix bleach with ammonia. That can create some toxic fumes. But what we've got here is something interesting that everyone's heard of pH. The question is, what is pH? And I made another video for my students, and I explored some of the really essential questions and really big ideas that this simple kitty demo gets at. And I really like this demonstration because it looks simple, magical in some ways, but the more you dig into the science, it really starts touching all kinds of areas of science. And so the other video talks about that. One thing about acids and bases, they're only things that include, that happen in solution, in water. And I can sort of demonstrate that a little bit. So this is my acid-base indicator in just water, and I'm going to dissolve sugar in it. And acids and bases always are water with a compound dissolved in it. So let's take some sugar, put it in, and we'll dissolve it and see what happens. Huh. It's dissolved. I don't see any crystals anymore. But nothing happened. Not all compounds, not all substances dissolved in water create acids or bases. Only some do. And so this property of acids and bases is interesting. It's been useful for thousands of years, really. It's used in all kinds of ways. And we'll look at that later. I'll show you, I'm going to put a different chemical in. This is actually the vitamin C. Let's see what happens when I dissolve that. Oh, look at that. I do get a nice color change there. So some compounds dissolved in water create acids and bases, or bases, and some don't seem to do anything. So there is a lot of interesting science you can delve into through even this very simple demonstration. Well, the question I do have, and you might have the same question, is who cares? I mean, why do we care about pH? Well, what's interesting about pH, which is a scale that we use to measure acids and bases, is that it's not a new observation or new science. The measuring is, the precise measuring is, but not the science. In fact, it goes back years, hundreds of years, thousands of years. Because for thousands of years, people have recognized that there are properties of acids and bases that were useful in cleaning, in preserving food, in making colors and dyes. They didn't have acid and base as a concept that we have now, but they were very aware of this quality and this range. In fact, acids, at this end, were talked about by Aristotle, the ancient Greek scientist, philosopher, over 2,000 years ago. And he used the word acetum. And acetum means vinegar in Latin and the Greek root that he was using. And, indeed, vinegar, this is actually vinegar, is acidic. And it's a bit sour tasting as well. But that's where we get the word acid from the Greek and Latin word for vinegar, acetum. At the other end of things, we sometimes call these bases, and we sometimes call this an alkaline solution. And the root of the word alkaline comes from a different language, still a culture of science, and it's Arabic. And alkaline, or basic, literally means alkali. It comes from the Arabic word alkali, which means ash. Alkali means ash. And in fact, if you take, or roasted, or charred, if you take wood and plant material and you burn it, and you make charcoal and ash, and you soak it in water, you get an alkaline solution, and we now usually call that a base, but both words are used. And so people have been paying attention to this. Why is it important? Well, alkaline solutions we combine with other plant and other substances, namely oils and fats, and that's how we make soap. And some of you may have done that even, where you mix an alkaline solution with oil or some sort of fat, and you can make soap. So, and soap making's been around for thousands of years. More recently, right now, why do we care? Let me ask you a question. Have you heard of exoplanets? Think about exoplanets. So we're now starting to look out into the solar system, the galaxy, for stars that might have planets going around them, the way we have our solar system, planets going around the sun. And of course, we're very interested to know whether there might be life on those planets. And what is the one substance that we know, or that we believe, must be present for life? Do you know what it is? I think you do. It's water. And we think it has to be even liquid water. And it might, maybe gas or frozen, it's hard to say. But we're looking for water and liquid water on other planets. And acids and bases occur as a property of some substance dissolved in water. Life, the processes of life, the chemistry of life, happens in water. We don't actually know of any other place where life can happen except in solution, the chemistry of life. And so it's also more interesting in the sense that it has to happen in a certain pH range of water. And the pH determines what kind of chemical reactions can happen, how quickly they happen, what materials form. Are we able to make proteins? Is the reaction going to happen at all? Can we break down things? I think you probably know one example is when you eat food, right? You eat food and as it enters your stomach, it anticipates the food, and it produces acid, actually hydrochloric acid, and we think of it as dissolving the food. I mean, it's actually, a chemical reaction of the acid with the food to break it down, to release, to access the energy and the nutrition and the other things that you get from food. And, and interestingly enough, when you're finished eating and the food moves further down the pipe, so to speak, the acid drops. Your stomach stops making so much acid. And, I mean, many processes in you and in life. I mean, in your blood, the process of the red blood cells picking up oxygen and then delivering it to muscles where it's needed or tissues, and then taking away CO2 is also very dependent on the pH of the blood that those cells are moving through. And your body actually regulates pH. Different cells, different parts of your body can have a different pH. And it drives the process of life. In our environment, it's the same way. A healthy lake, for instance, with lots of fish and plants growing well, maybe we take the pH. Maybe we find it's, around a pH 7, which is what neutral water is, and that's good. And we go to another lake not too far away, and there's no fish in it, or very few, and the plants aren't growing, and we measure that, and it's a pH of, let's say, 10. When we test it, we get something like this. Well, some lakes do have a high pH, but naturally. But it also might be that, there's some pollution, or there's some way that human beings are messing up that lake. And if we want to be healthy, we need to bring it back to a more neutral pH of regular fresh water. And so that could be another good use of it. Our oceans also, through the process of greenhouse gases, CO2 in particular, are acidifying. And the ocean's actually becoming slightly more acidic. And that's a problem for many living things in the ocean, coral reefs and other creatures like coral especially. So we're very interested in finding out, what can we do to mitigate the acidification of the ocean so that the ocean stays healthy. But here's the thing. I want to show you how to make it, because I'd like you to interest you in possibly doing this yourself and exploring the science and see where it leads. So the indicator is actually made from cabbage, red cabbage. And red cabbage is actually kind of purple. And there is a pigment in cabbage, in red cabbage in particular, called anthocyan. And I think antho in Greek means flower, and cyan is blue. This is actually a little more purple. But this chemical, if we can, it's a coloring pigment in many plants. It makes roses red, for instance. I think it makes blueberries blue. I know it does. But we can get it out of the cabbage leaf and into some water and produce this homemade, excellent pH indicator. We could even create a scale from this, right, if we wanted to. So I'll show you how to do that, how to make that. And we'll go from there. There's a lot of how-to on the internet on this. And I want to say, I think most of it is not good advice. Or at least it doesn't get the results I like. I like colors that are very clear. I think you can see that this one is, it's not foggy. They're nice, clean looking colors. And a lot of the methods of getting the anthocyan out of red cabbage, on the internet, you end up with this cloudy mixture. You also end up with sometimes a smelly, cabbagey smell, which I'm not so fond of either. So I'm going to do it differently. Let's take a jar. In fact, a really good jar to use is going to be a mason jar that can handle a little bit of heat. When I made it, by the way, I used a really big one because I needed a lot. I did this with a lot of classes. And we also have some smaller mason jars. So you just need to decide really how much, I'll move this aside, how much you want to make. And but these mason jars are an excellent something to make it in. Also, then we can put a cap on them, maybe keep them in the fridge, where this keeps at least probably for a week there, all right? And I'm gonna, even though this is what I used for class, and this one might be fine too. This is a pint. The first one was a half gallon. And this is a quart, about a liter mason jar. Put it right here. And the cabbage leaves you can just peel off. If you want to use a knife, you could, although you don't have to. So if you're not comfortable using a knife, why bother, And especially if you're doing this with kids or something, you can just peel off the leaves. And you don't really need to cut this up finely. And I'll give you an example of that in just a second. I'll peel off some leaves here. All right, so there's some leaves. I want to get a nice big one. There we go. And again, if you wanted to, you could indeed use a knife. But you don't need this finely cut. So, about 1 inch square would probably be fine. Because really you just have to get it into the jar. So, that would be fine. But I'll tell you something. You don't really need a knife. And it's probably safer just to tear it up. And it doesn't really matter. I can show you why. The anthocyanin part is on the outside, the purple part. If I take this close to the camera, you can see the inside is sort of white. And there isn't any anthocyanin, any of that acid-base indicator on the inside of the cabbage. It's all near the outside. And so all we have to do is put some in a jar, leafy form, and we only want to fill the jar, first of all, not stuffed, not packed tight, but about halfway, because there's quite a bit of this base indicator in each leaf. So I'm going to fill this up about halfway, okay? And that'll probably be plenty. I'm not even going to put it all in right now. Now, what you shouldn't do if you want a nice clear liquid is what a lot of the internet says to do. One, don't put it in a blender, okay? If you put all this in a blender, which is I've seen on the internet in several places, and you just get this gray smelly mush. It is purple, and it will have the anthocyanin acid-base indicator in it, but it's thick and it's cloudy, and it actually starts to smell like cabbage, a little sulfurous, very quickly. So I would not use a blender. You just have to tear it. The other thing that's really important is we're going to pour hot water on here, and we're going to bring water to a boil. Again, if you're with kids, or you're not good in the kitchen, you want to be careful, boiling water, but you want to boil some water. And we're not going to cook the cabbage. I repeat, we're not going to cook the cabbage. All we're going to do is fill it about halfway, and then pour, though just off the boil, so this has just been boiled, and we'll put this very hot water over the cabbage. And you'll get about, so it'll be about half full with cabbage, and then you'll finish it off with the hot water. And that is enough to kind of steep the cabbage and pull out from the cells in the cabbage leaves the anthocyanin acid-base indicator. And all you have to do is leave this to cool. It'll probably take an hour, hour and a half at the most, and this will cool to room temp, and you'll have a great cabbage-base indicator. You do have to get, of course, the cabbage out. And the way I do that is, I'll show you. Let me use my bigger one, because this is stuff I actually made earlier. So I have this empty container, and I use a wide mouth funnel and a strainer. I put like that right in there. Now you can use any kind of strainer. You could probably use a funnel and a coffee filter if you don't have that. And so here's some I made earlier. You can see it's got the purple color that's come out of the leaves. and but it's all the leaves in there. So what I'll do here is pour this through the strainer, and that will catch the leaves. Not very many actually. Mostly they're staying right in the jar, which is great. And I can move that over. And now, there you have it. Cabbage juice indicator. Cabbage juice acid-base indicator. And a nice thing again about the jars is that when you're done, you can put a lid right on them. And these jars I have are made for canning, so they can take the heat, and they're also easy to work with. So if I wanted to save this for another day, I could just take my little lid on and screw it down. And it's also easy to pour from. So my first choice would be one of these type canning jars, ball jars, canning jars. And my guess is you're probably going to want to go with the 1 quart size, although I went larger, and they even make smaller ones, right? All right, there we go. That's cabbage juice indicator, how to make it, and a demonstration.